And okay. All right, Mike, are you ready to be introduced?
I am. I'll close windows while you talk so we don't have any unexpected crashes here.
Okay. All right. Well, thank you guys so much for joining this meeting of the Spatial Biology Association. Today, our keynote speaker is Mike Snyder, who is the director of the Center for Genomics and Personalized Medicine and the chair of the genetics department at Stanford University. He's a member of the American Academy of Sciences and has been named a high impact and highly cited scientist for multiple years. He's also the co-founder of 9 companies that I know of, unless you've started one recently, Mike, and has also authored a book called Genomics and Personalized Medicine, What Everyone Needs to Know. He is a leader in the fields of functional genomics, proteomics, and personalized medicine, and is a major participant in the ENCODE project and in HUBMAP. His lab also uses a variety of omics approaches to understand human variation and health. And he's demonstrated the value of personal devices for preventative medicine. And according to an interview I read, he was able to use device data to make an early prediction of a Lyme disease infection, which is pretty cool. Today, he has agreed to discuss his lab's approach to spatial biology. And with that, I will turn it over to Dr. Snyder.
All right. Well, thanks very much for having me here. I am going to tell you about our work from the HUBMAP Consortia. And this is really, I'll focus on the spatial part, which has really been pioneered in our lab by John Hickey, who's in Gary Nolan's lab, works closely with his lab. Will Greenleaf's and ours all work very, very closely to map the human intestine in healthy folks. So, and these are the 3 main people who have been driving that project. Just super quick background. I'm sure most of the folks on this call already know this, but the work I will tell you about is funded by HUBMAP. It's an NIH common funded project. There's a link if you want to learn more. And really the goal of this program is, as it says here, it's to develop a framework for basically building single cell maps of organs. And there's technology development, all sorts of activities as part of this consortium. It's a very large number of groups all working together. I lost track of how many organs they're doing now, but it's somewhere between 8 and 10 or something like that. Different organs that are being mapped. I'll just also mention that we are mapping the human intestine, both small and large intestine, as you'll see in a minute. And we have a very complementary activity, which I won't have time to talk about today, which is to map basically early stage cancer using something called familial adenomatosis polyposis, which are folks who have an APC mutation. They develop lots of polyps and they're thought to be a great model for very, very early colon cancer. So a very complementary project there. And what we're doing for HUBMAP, we think is a terrific ground reference, if you will, for the cancer project that we're doing for H10. But I want to talk a little mostly, and since there's not huge amounts of time, just about the HUBMAP project. And as I say here, what we're trying to do is map the normal human intestine. Basically, for the most part, we've been doing 4 samples across the large intestine. So the intestine, you may or may not know, you're from your stomach, you feed into about 20 feet of small intestine, and that then feeds into the large intestine, and which sort of looks like this, wraps around the small intestine. This is about 7 feet, depends on the person, their age and such. And so we'll grab sections from this region, and I guess this is the ascending, transverse, descending, and then this is called the sigmoid region. So we'll take 4 regions here. Initially, we were taking 4 regions from the small intestine. They're listed here. And then more recently, we're taking 15 to try and get higher resolution, although they're coming out reasonably the same. So those 4 will probably be a pretty good representation. And so we've completed, if you will, a fairly detailed, you'll see CODEX analysis for spatial as well as single cell RNA and ATAC-seq across these different regions, which is building a first generation, if you will, map of these different regions. And so our core spatial technology is CODEX, because that's been pushed here by Gary's group. And for those of you who don't know, what you do is you stain with a small number of antibodies sequentially, and you can go through and basically by doing these repeated cycles, you get on the order of about 50 different proteins stained spatially, and then you can aggregate these images and build these beautiful maps. So you have to spend a lot of time getting a nice marker panel, and John's done just that. He's built a marker panel, if you will, for the human gut, 54 different proteins that map out a wide variety of immune and epithelial cells and such. And these are just a few examples shown here. And then the goal is for those of you who do this sort of stuff, as you know, you do these stains, and then you want to start assigning cell types. And so he's done that. This is sort of what the cell types look like. And then in the analysis I'll describe, we're trying to identify individual cell types. And then, oops, something got lost here. May have accidentally deleted something. But anyway, we're, oh, there it is. Sorry. And then we'll assemble these cell types into neighborhoods. And then you'll see even a little later, we'll make neighborhoods of neighborhoods to build communities to try and understand, the organization of cells in this complex tissue. And so that's really the goal. Now, it turns out it's a bit more challenging. It's quite challenging for those who do this sort of thing to make these cell types assignments, true even for single cell data. How do you decide what cells you have? And so we were doing this initially manually, and it would take about 25 hours per sample. So that's quite a bit of time. And then you think about it, you start piling up tens, if not hundreds of samples. That's a lot of person hours spent to just identify the cell type in each of these samples. So we've come up with an automated way of doing this. It's based on some work from Yuri Lesovic's lab. Maria Babrick from that group came up with a machine learning, a deep learning method, I should say, it's a bit supervised, you'll see in a minute, for trying to actually classify cells. We did it for cells, she did it for images. And this is a simple way of explaining it. If you have a known set of annotated images, in this case, animals, you can then go get a new set of images and you try and learn from this to predict this. And so that is very doable. This used to be challenging, now it's quite doable. And so, you see elephants here. We're in this labeled set, leopards, or is that a cheetah? I don't know, probably a cheetah, I guess. And octopuses, and so, or octopi. And then you can classify these in the unlabeled set. But what's special about what she did is you'll have leftover images that don't really fit so well into this. And then they'll be novel, and you can group these into their own class. So you can actually then define new cell types. And that actually works. So this is another way of representing this. You have all your labeled cells, you have your unlabeled material, and then you basically, based on the information here, you can assign labels to a lot of these ones and annotate them, and then you'll have the leftovers and you group them into categories, and that defines your new cell type. And that actually works quite well. So at the end, you'll wind up getting all these labeled classes and generate clusters of, we'll call them unseen classes. And it turned out just saves a ton of time. It's not that you don't spend some manual time looking over these to make sure things look good, but it does work. And so basically you can do this. This is one good example. You can do this across, for the same individual, you can go across different regions. So here is one, this just shows it works on the same image, but you have an annotated sample. And then you can assign it, I should say the package is called Stellar. The acronyms back here, spatial cell learning. Yeah, and you can then label the next, this shows on the same one that it works, but you can go across different regions of the colon, and we can actually label. So if you say we have 4 tissue regions, you can train on 3, and then you can assign labels to the 4th region on the same person, and it works pretty well. And you'll have those novel cell types if you're in a new region as well. And it also works across individuals. So you can basically go from one donor to the next, and so on. So you can basically fairly quickly annotate all these samples. So this really has turned out to save us a ton of time. So any of you who do this sort of thing, you're more than welcome to use this. The preprint on this is actually should be up on bioRxiv, and we're sharing the algorithm and such. So again, you can assign labels in a fairly automated fashion. And I think the same principle should work for other kinds of data. We've just set it up for CODEX right now. So using the system now, we've begun to map out the human intestine. And as I said, we're doing 4 regions from the small intestine, 4 regions from the the large intestine. The large intestine, for the most part, looks pretty similar to the different regions. We didn't know that going in. There are some differences, but they're much more similar. The small intestine has a lot of variation across it, which is kind of interesting. And so we mapped a total of 1.2 million cells from 25 different cell types across these 8 regions of 4 different donors, so 32 samples in total, and annotated all these cells. And the first conclusion, which I don't think will surprise anyone, I have to hide people's pictures so I can see this myself, is that, you will find differences in cell present cell types across different regions of the chromosomes or intestine. So going from, this is coming out of the stomach into this first region, small intestine, then you move into the colon over here. And so you'll see the stromal cells, for the most part, seem to be kind of similar numbers across the different regions, but as soon as you get into other cell types, immune cells, epithelial cells, there's a lot of variation. And just as an example, this is an average, and we can show these are statistically significant. The CD8 cells, which are pretty interesting, they're pretty abundant in the small intestine. They start getting less abundant in the ileum, which is near the colon, and then less in the colon. And here's another good one here, the enterocytes. They're absorbing cells that they're very abundant in the small bowel, small intestine, and then become less abundant in the colon. And then we have other things like this class of enterocytes. They're basically less in the small intestine and more in the large one. And so you can start quantifying all this. And visually, everything we see kind of backs up what we see here. So the other kind of interesting thing is you can start asking, well, are the cells, when they're in these neighborhoods, are they sort of in the same density, or are they more spread out? And this actually could be relevant to their biology, and the part I'll tell you next, the neighborhoods. And you can show actually the density of cells will differ a little bit depending where they are. So for example, plasma cells are generally the same density, if you will, no matter where they are in the colon. And other cell types like these CD8 ones, they're actually a little different density depending where you are in the colon, and this relates to their function, probably to pick up strange antigens and fight them at different regions. And so they have a very different distribution, if you will, across these regions. So one of the major goals of doing spatial mapping in general, it's fun to see these cell densities and all this sort of stuff, but it's really to see if we can understand cell context. And so we've been taking these individual cells, grouping them into so-called neighborhoods. And so here are the individual cell types listed across the x-axis. Each column is a different cell type that we have markers for. And then over here, we can sort of group them into neighborhoods. And so the outer follicle pretty much has what you would expect. It has these sorts of immune cells here. The inner follicle is pretty similar, although the distribution of cells can be a bit different. It tends to have more B cells and less of some other things. These tend to have more of the CD4 cells. So if you're an immunologist, this stuff is really pretty fascinating. You may or may not realize most of your immune cells are in your gut. So it's kind of a pretty important area actually for your immune health. And that's because it's getting exposed to all these things you eat and possible pathogens and plastics and pesticides and all that sort of stuff that's in your food. So anyway, not to scare you for lunch, which it's about to be here. But anyway, this is the kind of what you can see. That's the immune cells, but you can do this for all kinds of other things too. Here's stroma and innervated stroma and so on and so forth. So you can define these neighborhoods based on their cell composition. And you can do this in a more formal fashion by clustering analysis. And so we've characterized, if you will, 18 different multicellular neighborhoods across the intestine. And then, not surprisingly, these neighborhoods will vary across the intestine. For the most part, it fits with what you would say about cell types, but you gain a little more information as well. And as an example, here I mentioned before, CD8 cells are more prevalent in small bowel, less in the colon, and that reflects in these neighborhoods that we see with the CD8 cells. And but we'll start finding some other neighborhoods, that will be more colon specific or colon enriched, I should say. Like this is one here. Here's mature epithelial cells. They're much more that kind of class of neighborhood is more small bowel and so on and so forth. So we can see what kinds of neighborhoods. And again, these will be defined by specific cell compositions. And then, there's a lot of sort of interesting stuff, like are the neighborhoods themselves conserved in their composition as you go across the intestine, and some of them are, and some of them are, and some of them aren't. So the alar follicle actually varies quite a bit depending how you go across the intestine in terms of how it's its cell composition a little bit and its neighbors as well. The other thing we can do, and this got complicated, I'm sorry, I just got these slides last night. I didn't have time to simplify them. I got through all of them but this one. But I'll explain this one and verbally listen to what I'm saying. So what you can do is you can take these neighborhoods and you can say, well, are the neighborhoods themselves in neighborhoods? So how do they associate with one another? So you can take it up a notch. And it turns out there's a non-random distribution of neighborhoods as well. And we're calling these communities, sorry, a lot of typos here. As I said, I didn't quite get time to deconvolute this. And probably the easiest way is to just look at panel C. I think you can see this, where you have cell types that will assemble together into neighborhoods. And then these neighborhoods themselves can assemble. You can see that different types of neighborhoods prefer to be with one another to form communities, or we can even call them major communities. Not quite sure what the right nomenclature should be after this. These could be small communities and larger ones, I suppose. But the point is that we can actually see which of these like to be with one another. And then there's a lot of interesting biology that assembles around here when you think about epithelial function and secretion, as well as immune cell function, which is kind of a big deal, that these immune neighborhoods actually do differ across different regions of the intestine. And we think that's important for their function and the way they capture and present antigens is one example of the kinds of information you can obtain from better understanding the organization of this. So the other thing we've been doing, and we do a lot of this, as you might imagine, we do single cell RNA and single cell ATAC-seq. And these days you can do it on the exact same cell with a technology called multiome. And so we've done just that. We've done single cell RNA and ATAC-seq. I should call it single nuclear because we're dealing with the nuclear parts. And when you do that, we've done it for the exact same tissues I mentioned, all 8 sections for the 4 donors in the same regions. And you can start, clustering the cell types, and this is no surprise. Here are the stromal cells, the immune cells. In this case, the epithelial cells, the colon shows how different they are. We'll segregate, the colon ones will segregate out from the bowel. And then what you do is you go into these, for those of you who don't know, you go into these populations and try and further substratify them by getting rid of the rest and seeing, well, how do these compare with one another? And here's an example of the immune cells. And so if you just take the immune cells and say, well, how do they relate to one another? This is sort of a similarity score. Again, it's called HUBMAP for those of you who don't know. It's a way of, it's like PCA a little bit, but it's not exactly the same, but it basically groups cells based on their in this case, RNA relationship. This is RNA data. This is ATAC data. And you can see, you can group them, and we can annotate them by various ways, again, using known markers. And there, I'll come to that in a little bit. And just like for the CODEX data, you can show in probably even a more quantitative fashion how cell numbers will differ across different regions of the intestine. So once again, well, in this case, it's macrophages, I guess, are showing up more in the colon and less in the small intestine and so on. And these are the stromal cell population, same things, they'll show shifts as well. And for the most part, it matches pretty well. But the other nice thing you can do with the ATAC data is you actually, because it's high resolution, you can even get in there and start defining subpopulations. So we came up with some things we called secretory unknown 1 and 2. And we have a variety of subclasses and enteroendocrine cells that are that we can define. And some of these are known already in the literature, it's been defined, but some of them seem to be new. And so what you do, for example, mucin 5 and mucin 6 cells are found in the small intestine. And what we've been doing now is we're going back and getting very specific probes of mucin 5 and 6. And we're going to see if do they have the same cellular distribution, or are they in different locations? We know actually, by the way, they differ in their abundance across different regions of the small intestine. I don't have that slide here, but we don't know spatially if they serve the same role or how that works. So this is the kind of thing you can do. So adding single cell RNA and attack data onto the CODEX data really gives a much more, if you will, high resolution view. And also, I should point out, we missed one region in the CODEX data because we didn't probes for it. And there were 2 cell types in the CODEX we missed with the single cell data because they just weren't that abundant, so they didn't show up in our cluster analysis. So the approaches are extremely complementary, and together, as I say, give you much more resolution. They create hypotheses that you have to go back and test. So we'll be making, we have antibodies of mucin 5 and 6 as an example, and we'll go back and stain with CODEX to see where they go. And the same thing with all these endocrine cells. We're very interested in their spatial organization, how and are they really differ? And there's going to be arguments that any of you do this stuff. What's the difference between a cell type and a cell state? And you can spend your entire life debating that probably, but something the whole field wrestles with. The other cool thing you can do when you integrate data like this, I see a better wrap actually, is you can get, again, deeper resolution data. So from the CODEX data, I already mentioned we can define neighborhoods and what cells are near each other. You can do this very systematically. And so these are certain, for example, plasma cells are next to enterocytes, they tend to be enriched. And then from the single cell RNA data, you can go into those same cell types and say what receptors and ligands are in these, and therefore might actually be responsible for communicating across these nearby cell types. And this is just one example. I think we came up with 47 or some number like that. That's statistically significant. But this will show one example. This is the work from Almudena Perez and Sylvia Plavidis' lab. What she's been able to do is do what I just said. So for example, here's this ligand semi-4D on plasma cells, and here's a receptor. This is all based on sort of paralogs, if you will, that's in these Ta2 cells. And so this sets that maybe this is based on known data related ligand and receptors. This may be how these cells talk to one another. We can make these molecular communication hypotheses, if you will, or nominations for cell interactions from this. And so this is a sort of way you can actually try to get to even a molecular map. And again, this will need to be validated with more data. So these are the folks who did all the work. John Hickey did all the spatial work with CODEX in Gary's lab. Stephanie Nevins and Aaron Horning were major leaders in the project in our lab, and Winston Becker did a lot of the single cell analyses, and Almudena and to some extent Alice did a lot of work in Sylvia's lab. So together, I think we formed a very complementary team to try and take on this tough problem. And I did show a picture of John just because he did a lot of the work. So anyway, I think I saved some time for questions. I hope that's about the length you were hoping for.
Absolutely, yeah. Does anyone have any questions for Mike? And maybe I can start off with one.
Sure.
Okay, so I thought it was really interesting how you mentioned just the effects of environmental variables and how much variation there can be in intestinal tissue. And I was wondering kind of what your mindset is to capturing that, what kind of donor numbers you think you'd ideally need to kind of capture that variation.
I don't know the answer to that. At the level we're mostly just trying to make general descriptions across the colon. You know, there's age, obviously there's ethnic group. I didn't get into this, but one individual was African-American. The other three are white. For the cancer, we're fortunate because where we are, I guess kind of sad, but there's a lot of Hispanics. So we just need to capture more from ethnic groups. That's obviously a big push in HUBMAP and other groups. Age is a big factor. The thing about the colon is I think diet would be extremely interesting to look at as well, given that people's response to different diets. And in the long run, I think we want to understand how some of these things, pesticides, certain things are pretty common exposures that you actually will get not only from airborne exposures, but from, ingestion. And so I think getting that kind of information, how that affects the structure of your gut, will be very interesting too. So like all things, the laundry list can go on and on. But I think for normal, healthy diversity, I'm not sure what the right answer is. Let's just start with somewhere between 10 and 20 to get a reasonable reference map and take it from there. That's sort of how we're thinking about across different ethnic groups and ages. So, and we may be able to impute stuff. Right now, it's very expensive if we're going to try and do serial, which is our next step. Build really spatial serial section maps, and that will cost quite a bit with current technologies. So.
Thank you. Yeah.
There's a hand and some questions. I'll let you field this actually.
Okay. Well, Clarence, do you want to ask your question and then we can get to those in the chat?
Sure. Hi, Mike. It's Clarence. Great talk. I just want to clarify my understanding about the annotations that has gone into Stellar. So it's been expertly annotated on one donor sample. Is that right? And then it was transferred to a few other donors to assess the, how well it performs on samples that it hasn't seen before. How do you assess that? I'm reading the preprint and it says you look at the average marker expression profiles. Can you comment a little bit more about that?
Average expression. Well, yeah, I guess what we're doing is, to be honest, I thought we had done 3, and then I know for the purpose of the paper, we showed proof of principle. We could do 1 and go across different regions of the same person, as well as go across people. We've further validated, I don't remember if it's in the preprint or not, by doing 3 people and testing a 4th and got pretty much the same results. If you're going to get into the CS, deep learning side, what you do, you mix the features together, and then we're going to cluster the unknown ones with the known ones, right? We label these things as the known things as features based on, I presume, their expression values across, all the measured transcripts, and have a similarity score across all of those individual features, and then define that as a cell type, what they have in common. And if you're going to ask me the math behind this, I'll have to put you on to Maria, to be honest, from Yuri's lab. But basically, that's the general principle, right? You're defining the features that make a certain cell type, and then trying to assign based on closeness other features. But it's all done through a deep learning system, so you don't necessarily know all the individual parameters that go into it, but you do know the outputs. Does that make sense?
That makes perfect sense. But so there aren't expert annotations on the other data sets that it has not seen before, right?
Well, at the end, the colon's pretty well characterized. So we go back and look. You do put a manual component on top at the end after you've done all your assignments, and you try and see if you're missing anything. So I think, I do think I may have simplified it. There definitely is still a little bit of manual time, but it's just nice to have most of your assignments already done. I don't think we're missing anything totally clearly, but I think we're more missing things because we don't have the right probes, if that makes sense, than the way we're doing it by machine learning. So meaning the RNA-seq data told us, well, there's some subclassification we didn't have. So if I were to spend energy, I'd be spending more energy getting the RNA-seq data to use to better define cell types and states, and then go back and stain it. It seems to me we're going to get more novelty from that sort of thing. Does that make sense?
Yeah, great. Thank you very much, Mike.
Okay, so that's how we approach it. Yeah. And we have a new way too of, this might relate to the Kenishi's question. Let me see, it's a collective protection. Oh, how thick a frozen section. I don't remember for sure, but I think it's somewhere on the order of 5 or 10 microns on the thickness of the section. You can send me an email on that and I'll have to pass it on to the other folks. But that sounds right to me, 10 microns, as I recall. Might be 5. Okay.
And then obviously people who put their questions in the chat, you can feel free to unmute and ask it
Oh yeah, I
yourself.
should have let you ask it. Sorry, you can ask your own question. Hopefully I answered it right. Thanks, Angela.
Yeah, Amit, do you want to ask your question?
Yes. Hi, Mike. This is actually Amit Custom from now at Georgia Tech, but now I was previously at Gary Nolan's lab.
Yeah.
And John is actually.
So you know more what I presented than I do, probably. All right.
But thank you so much. This was a great presentation. And I learned a lot about the gut health as well. So my question is actually regarding your vision. So I heard another talk about personalized medicine where you wrote all these cool gadgets and the track digital health. Oh yeah, those ones. And also this talk, right? I'm curious, how do we put these results together, right? Can we integrate?
Well, we're very keen on that. So we measure pretty much everything we can. I wear 8 wearable devices every day. And I think as relates to it's trickier for this because the gut, you just don't take biopsies off people's guts all the time. So, but you do, blood cell draws all the time. I do them myself quite a bit. So I think we can relate a lot from the blood back to gut health, and then and the microbiome in particular. I do think in the long run, I think an area that's very understudied at NIH is nutrition. And I think they're aware of it because there's been some new nutrition initiatives you may or may not know. Because, if you think about something people eat every day, and food really is medicine. And given, you may not realize something like 10% of the population has irritable bowel syndrome. And of course, inflammatory bowel disease, Crohn's, these are conditions that are very much on the rise. So, and they're very, very heavily influenced by your immune system, which is very heavily influenced by what you eat. So I just think this whole nutrition, gut metabolome, immune, that 4-way axis is so important. And I don't think you can understand one without understanding the other 3. And I think genetics and all these other, chair genetics, of course I'd say this, but I think they're all going to play in there too. But minimally, we've got to understand, the nutrition, gut, metabolome, immune axis, because they really are. They're just so interdigitated. And so I think as you start perturbing, just think of where we are now and just think about we pick on the pandemic, but there's a bigger endemic out there. 9% of the population's diabetic, 33% are pre-diabetic. Those numbers are skyrocketing. And a lot of that's nutrition-based failures, if you ask me. People eat, typically about 12 grams of fiber a day. We should be eating, but people say women 25 grams, men 35 grams of fiber. So we're not even close to what we should be doing. So I think we've got to understand all this and then, implement the right, basically mitigations. And because of this, a lot of people are dying during the pandemic, right? Who are diabetic and obese. So one of the most vulnerable classes outside of age is actually people's diabetic and obesity status, BMI status.
Thank you. Yeah, I think this is great. Both visions, hopefully.
Yeah, well, that was a little bit of me standing up on a podium, preaching, I guess. Sorry about that. But anyway, all right. It's something I feel passionate about. It's probably why we study the gut.
And Kenichi, did you want to ask your second question?
Oh, yes, please. If that's okay. Yeah. So could you briefly comment on how you are convinced about looking at the 2 different data sets, sequencing and imaging, and how you are convinced that these 2 populations are the same, particularly when like it's not simply defined by cores, like a few biomarkers, but like if you look closely, particularly in the sequencing data, probably it's a bit more context or it's a subtlety that you would have to think about. So is there any simple way that you could come up with, or
yeah, I mean, you're right. It's all kind of manually done, right? We see a marker on CODEX and we see this set of cells in the RNA-seq population highly expressed. And so we assume they're the same. Now, again, there's usually more subcellular or population resolution in the RNA data than there is in the CODEX data. So we can define further cell types. So I guess you'd have to call it a preliminary assignment. It's kind of where the field is stuck, right? It is what it is, because the RNA and protein don't have to be perfectly correlated. We're very aware of that. I think in the immune populations, we're not so bad because we've done plenty of, single cell and FACS sorting. So that one's pretty good. I think in the epithelial, there's a lot more to be done, especially for the subpopulation assignments or subgrouping assignments. So it is what it is. We're just making the best correlation we can. And most of it's manual. So I don't disagree that that leaves you prone to problems. But it's not unreasonable. And to be honest, I find the whole field, that's one of the reasons I like this deep learning approach that we were doing for the assignments, because it is a more systematic method. And I started to say, but then I forgot, we're trying to use now ATAC and RNA data to better make cell assignments too. People typically do RNA separate and ATAC separate, and then say, oh, I think they kind of line up. But same thing, very visually, very manually. And I think we need more systematic, quantitative measures for deciding this. And my own view is that this is ultimately how we'll define what is a cell state from a cell type. It's all going to be by closeness relationships. And it'll probably depend on, to some extent, at what level you want to look at things too. So that's, I think getting quantitative measures around all this stuff versus, oh, here's what I see and here's what I manually do, it will be important for the field. It's a great question.
Yeah. Thank you so much.
All right, guys, we have time for about one more question, if anyone has one. Well, I guess maybe I can ask one more question.
Oh, you do that.
Fire away.
Okay, well, I guess I was curious whether you are or are interested in integrating sort of a metagenomics approach, looking at the bacterial populations in your intestinal tissues as well.
Yeah. Now, one tricky part is that, so totally, because we do a lot with the microbiome in general. And one of my gadgets, just grabbing this, actually measures airborne exposure. So we're very interested in relating, and we've done this related sort of what people are breathing and ingesting to gut and metabolic health. So it's an area of very high interest. The one caveat we have is, at least for our cancer patients, and to some extent for our HUBMAP donors, are actually organ transplant folks. And so they've been kind of cleaned out, if you will, of the microbiome. So we're taking whatever we can find there. There's a little bit of residue. It's not ideal. So it is the case, it'd be lower quality. We do that because then the quality of material is really, really high from these organ donors. But the microbiome is probably not so native. Whereas if we could take, I guess it sounds gruesome, but people from car accidents and things like that and got their colon there, we get a better representation of the microbiome. Now there, you'd of course love to know what they were eating before they had that car accident. I don't know how easy it is to get that. So there are challenges here, is what you can see. But anyway, very much want to know that, because it comes back to what I was saying before about nutrition and the microbiome, because basically, your food feeds your, especially in the colon, your microbiome. And so that's going to be really, really critical, I think, for the organization, especially the immune organization of the colon and whole intestine for that matter. Great question.
Thanks. Okay, and I guess just one, would you have any, I guess your audience here is all junior investigators who are interested in spatial biology. So sort of before we let you go, do you have any advice for people who are looking to get into doing these technologies and kind of looking forward into where the field is going?
Yeah, I mean, I think most of what we do now is it's Neanderthal, quite frankly. So much like I said about this annotation method we came up with, I think there's a zillion ideas out there. So I think as a new investigator, take one of these things you hate doing, figure out a better way to do it, and then make it available to everyone else in the Zoom call, and you'll be a hero. So I guarantee there's a zillion ideas out there. You have no idea how many people are hitting their head when PCR first got published. It was like duh, I could have thought of that. And I guarantee there's a zillion other ideas out there like that. So fire away. You got to figure out how to get them out to the community, though. Some people really use them. So
Awesome.
that's from the technology side.
Yeah, thank you so much for your time, Mike. This was an amazing talk and great discussion. And of course, you're welcome to stick around. But as an association, our next agenda item is going to be to discuss what workshops we'd like to do. So
I might
totally
jump off for that. Anyway, any questions any of you have, happy to talk to any or all of you. Feel free to reach out. Bye all.
Thank you.